an optimized NGS data analysis approach, the Genome Station experience. So we'll we'll talk about the genomics revolution. How exactly did this all pick up? So in the year 2000, a press conference was held in White House to announce an extraordinary feat, which is the completion of the first draft of the human genome, which later, as we all know, got published in the year 2003. This was a landmark experiment in the history of mankind because a lot of scientists from 5 different countries, 15 different research institutes collaborated to decipher the mystery that was the human genome. And again, for the very first time in the history of mankind, we were able to read all the letters that constituted the entire human genome, 3.2 billion to be precise. And if we talk about the progress that has been made in the field of genomics since then, well, instead of 13 years that took us to sequence the first human genome, we have brought down the time to around uh, say 3 days, and the cost of sequencing has come down from $3.2 billion to around $1,000 per genome. Now, this experiment kind of acted as a precursor to all the developments and advancements that took place in the field of genomics and sequencing technologies in general. And if we talk about the current day and age, this technology has become an integral part of all the modern research that is happening in the life science sector. Post this experiment, there was a flood of genomics data. Not only humans or mammals, to be precise, a lot of genomics based experiments began to be conducted in the field of plants and specifically crop and plantation crop research. So if we talk about the amount of data that this entire domain is generating, it is supposed it is expected to surpass YouTube and Twitter as the single largest source of data generation in the world, and it is expected to cross around 40 exabytes of data. If we talk as layman's, our imagination as far as the data size is concerned runs into TBs, right? Here we are talking about something 3 times, 4 times more bigger than that unit. We are talking about 40 exabytes of data. Similarly, it has been also estimated that by 2025 we are expected to sequence around 1 zettabyte, which is around 1 trillion billion bases per year. So you can very well imagine the amount of data which is going to be available out there. And it has also been estimated that there would be more genomes sequenced than there are people on the planet. And you can very well imagine the amount of data. Again, I'm stressing on it, the amount of data that this particular process is going to generate in the coming time. Now, if we talk about a standard sequencing approach, there are two main approaches that we apply whenever we are trying to plan a sequencing experiment. One is a de novo sequencing approach, which is characterized by the absence of a reference, and the second one is a resequencing approach, which is characterized by the presence of a well characterized reference genome. And this approach applies to whatever molecule you are working on, be it DNA or RNA. Now, if we talk about various applications that are being worked upon in the field of genomics, you have whole genome sequencing, which covers both de novo as well as resequencing approaches. You have transcriptome sequencing, wherein you sequence the mRNA content of a particular species in order to get an idea of gene expression. You are talking about targeted resequencing, wherein you specifically target certain genes or regions which you sequence, right, in order to, most common example being the exome sequencing approach. Then you have genotyping by sequencing, which was very aptly explained by Dr. Awad in yesterday's session. Then you have chip sequencing, which deals with DNA protein interactions. You have metagenome sequencing, a lot of which was discussed by Dr. Paul Raj in his session. You have small RNA sequencing, and you have bisulfite sequencing, wherein you study DNA methylation patterns. So these are some of the most commonly utilized applications in the field of genomics as such. Now, if we talk about the tools that are available in the field of genomics and data analysis in general, because once you generate the data, obviously the data will not present findings automatically in front of you, right? You have to analyze it and make sure that that you are able to churn out something meaningful from it. So if we look at the stats between the year 2020 2021, the time when most of us were in most of us were under lockdown, self isolation, right? 325 new tools for various genomics data analysis based applications have been designed and published. These tools cover various applications, right from alignment, wherein you compare your data against a reference genome, to assembly, wherein you arrange your data to form something meaningful, which mostly is a genome. So tools ranging to all these applications have been designed, developed, and published. We are talking about new age alignment tools like Sibelia Z, Callisto, Salmon, to more sophisticated hybrid genome assembly tools like Wengen, WTD, BG2, and Shasta Toolkit. And if we talk about every application out there, the list of sophisticated and advanced tools that are coming up is ever increasing. So it obviously is the need of the hour for us to upgrade our analysis pipelines and keep them abreast with the latest that is out there in the research world, right? Because sticking to a fixed pipeline will no longer will start making sense. So you have a pipeline fixed right now, most probably by the next 2 years using the same pipeline to get your analysis done won't make sense. There will be better, more sophisticated tools to give you more insight into the complex and dense data that is the genomics big data. Now, all said and done, the biggest advantage of all these tools that are coming up is most of them, almost 90% of them, are open source in nature. When I say open source, I mean these tools are available, and you can download them, you can utilize them, you can improve on them if you have the right skill set. Now, all said and done, we have talked about the brighter aspects, wherein we discussed about various applications that are there in the field of genomics, the amount of data that we are looking to generate, uh, and all these open source tools which are available to us to analyze the data that we are generating. But there are problems which most of us as biologists face on a daily basis. The biggest of them is, as of now, your DNA sequencer does not produce a complete genome, which your researchers, which we as biologists can read like a book. Rather, uh, it churns out data in the form of shreds, like shreds of newspapers without any organization of the fragments. You have to arrange them on your own, right? Again, the data is humongous as far as size is concerned, size and volume is concerned. So you are not expected to analyze it or work on it manually. You ought to have tools, and tools which are backed by the optimum hardware to analyze and interpret those data sets. In addition to this, again, the amount of data that we are looking to generate, both in terms of the output that we are get input that we are providing to our tools, and the output that those tools are generating, we need to have state of the art storage devices or mechanisms which can accommodate that amount of data. We cannot simply depend on these classical sources of data storage which we have been used to, which we are accustomed to, most of the times, like pen drives, internal hard drives, or external hard drives, right? We ought to have sophisticated state of the art solutions available that can help us to store the data and retrieve it whenever required. And this is the harsh truth as far as the informatics part is concerned. Sequencing machines have improved and have started to appear in more and more laboratories in the country, but the same cannot be said about state of the art infrastructure for computational analysis and data storage. That is severely lacking in most of our premier research institutes as well. So, there is this reason behind this skepticism. People, researchers, they are interested in procuring resources that can help them crunch their genomics big data into something meaningful that can add value to their research. But there are few reasons that make them a bit skeptical about taking the plunge and setting up a computational facility in their own local environment. The first and foremost is the inability to figure out optimal configuration for their establishment. most of the times we end up overspending or underspending, and the setup that we procure does not cater to our requirement to its full capacity. Again, most of the tools that we had discussed previously, the latest one or the previously available ones, all of them are, in fact 95% of them are command line based. And there's a general fear of the command line, the black screen and blinking cursor in the mind of us biologists, right? So we refrain from going into that direction, just because we are not that comfortable with the Linux or the open source environment and the command line as such. The third and biggest challenge is who is going to put this facility into use, because the command line, the fear of the command line is so overwhelming that most of us, even if we procure a facility, even if we have a computational setup in our lab or in our establishment, we end up losing interest because the fear of command line is too overwhelming, right? We have commercially available options as well, but there are quite a few players in the market that provide us with various toolkits for genomics data analysis. But their biggest two of their biggest limitations is: a, they are expensive, and b, they are rigid in terms of the pipelines that they accommodate. Expensive in what sense? You have yearly reoccurring license agreements. On top of it, these tools do not cover for your hardware requirement. So it becomes all the more difficult for us to procure a system, because you have to pay an exuberant amount to renew your license for that particular tool. On top of it, you have to invest separately in your computational facility as well. Okay. And rigidity, because you cannot add and modify the existing pipelines. Many times what happens is your analysis requires a non-standard approach, right? And for that non-standard approach to materialize, you need to mix and match your tools accordingly. That is unfortunately not the case with most of our commercially available toolkits for data analysis. So till now we have discussed about the problems that most of us face uh, when we look to take control of our data analysis and set up a facility in our establishment which we can use to do the same. Now let us talk about the solution. Welcome to Bionivet. We were established in the year 2011, and we are an ISO 9001-2015 certified company. If we talk about our various offerings, we have been actively involved in various uh, research based activities in institutes, with institutes of repute. We have in past and are still working with various DBT funded, CSR, ICR funded institutes, and we have been providing sequencing and analytics based services to them. Close to 80 publications are a testament to our hard work and expertise in the field of genomics and associated data analytics protocols. We also are into trainings and uh, education domain, wherein we conduct regular online as well as offline training programs for students, researchers, and scientists alike. During this lockdown, we have been able to train more than 3,000 students, and the audience was spread all across the globe. We had candidates coming in from countries like Costa Rica and Czech Republic, to name a few. We are also heavily into developing hardware and software based solutions for simplifying genomics data analysis and storage. That is what I'm going to talk about in the coming slides. That is what I'm going to stress upon in the coming slides. So we have developed products, softwares like SQIT, SQDB, and Gstack. SQIT is a simplified analytics platform wherein you can perform your analysis on your own without actually having to take the trouble of typing the commands on the terminal. You have a GUI, a graphical user interface, wherein just like you fill your regular forms, you can upload your files, you can analyze them, and you can make sure that your data is processed. We have an interpretation platform in the form of SQDB, wherein you can upload your Excel sheets and you can derive all the meaningful pathway related informations from the same. We have a hybrid cloud storage platform by the name of Gstack, which doubles up as a project management platform as well. You can store your data, track the proceedings of your project, and manage, share, collaborate data as and when required. Now, what all this comes bundled up in our state of the art, specialized, and optimized hardware solutions, which we have developed in collaboration with Dell India Private Limited, the Genome Stations. Now, as a solution, what do we propose here? We discussed about all the problems. So, what do we propose here as a solution? We propose a proprietary Bionivet product developed in collaboration with Dell, which boasts of an optimized hardware for better performance. We have made certain optimizations in the hardware, and we have observed 20% enhanced performance while handling genomics data. We propose a machine that comes with a customized operating system. We like to call it as the GSOS, the Genome Station Operating System. This operating system comes pre-installed with all the tools and latest data analysis pipelines which are out there, which are published, which are well cited. We pre-install all those tools in the operating system so that your machine becomes a ready to use plug and play device. A graphical user interface based analysis package, SQIT, which simplifies the data analysis for you altogether. Apart from SQIT, a system which is embedded with these simplified automated pipelines for NGS data analysis for various applications as such, and above all, access to a hybrid and dedicated genomics cloud storage platform, which is Gstack. So we present before you, as I said in the previous slide, the Genome Station. We have desktop level to server level solutions for the same, depending on your budget, depending on your analytics needs, depending on the sample load in your laboratory. We have systems suiting each and every need and pocket. So this is the Dell India and Bionivet collaboration white paper. It can be found on Dell India website as well, wherein this is a case study rather, wherein the Dell management has reported how these optimized solutions which we have co-developed have made life simple and more friend and made it more friendly for biologists who generally are not that much into command line based analysis, right? Now, see, there is already a lot of competition in the market, and we, as I have already discussed, there are a lot of customized pipelines available out there. What makes the Bionivet product, the Genome Station, stand out from the rest? Well, first of all, we provide complete hardware and software support for your Genome Station. Okay, any problem in the hardware, any problem in the software, in your operating system, in the tools that you have installed, we take care of that. We provide complete cover to your machine. We make sure that your system comes with all the required data analytics packages. All those packages come pre-installed in your system. As I said before, all you have to do is connect it to a power source and start using the system. From raw data quality control to gene network to finding variants, right, to plotting circular genomes, everything that you have in mind, anything that you want to interpret from your data, you can do that using the power of your Genome Station. Also, updates released over a period of 3 years will be plugged into your Genome Station free of cost. Also, we have 9 years of experience in genomics data analysis and close to 80 publications, as I had discussed below. So we also provide free consultation to our esteemed Genome Station customers so that they can utilize their systems to the maximum. Obviously, our analytics team with vast experience in the field of genomics make sure that you utilize the Genome Station to the best of its capacity, and we make sure that you put it to use and don't just use it as a regular system. Use it as a specialized machine for genomics data analysis. We also have on-site online trainings included with the overall package, so we make sure that we train all of you to ensure that you know how this machine works and how to make the best use out of it. We also help in sourcing skilled manpower with experience of working on genomics big data and high performance computational systems as well. So all in all, you can see that this particular product works as an end solution to all your problems related to genomics and genomics big data analysis, right from support to making sure that you are utilizing this facility to the fullest. We have it all covered, so that gives an advantage to this product. Moreover, as I said before, we have the machine comes packed with various automated analytics pipelines, so you do not have to invest separately in a toolkit and then separately procure a system altogether, doubling up your expenses. You don't have to do that. A single machine comes bundled up with everything that you require to make your data analysis simplified. So this is SQIT, which I was talking about previously. So this is a small command. It might not seem small because it is running into 3 lines and it involves a lot and lots of characters that we more often than not end up despising, right? So it's a very big command. It is for performing raw data quality control of your genomics big data, quality control being the first step of any analysis. Now if you look at this command, it involves a lot of parameters which at present all of you might not be able to understand, because obviously some of you might be new to this. And imagine remembering this command, writing it, and then finding out that once you press enter, a very silly error comes up on your terminal screen. It would frustrate you to the core, right? Now, imagine such kind of a setup wherein the same tools require you just to fill this form, the simplified form, not to type the entire command, but just to fill in these forms and click on submit. The moment you click on submit, your analysis is done and you get a customized reports bearing all the necessary information from your data. Definitely makes your life simpler. This is what SQIT does, right? From RNA sequencing data analysis to analyzing bacterial genomes to performing metagenomics based data analysis, this pipeline covers everything that you will need to make sure that you interpret your genomics big data to its fullest capacity. Again, we have several automated pipelines here in this SQIT, as I said, right from getting a list of your differentially expressed genes to various plots and figures that are associated with it. This is just for one publication, which is RNA-seq. We have similar sort of reports for which I'll be showing to you in the later stages of this presentation, that cover various other aspects as well. So right from heat maps to volcano plots, you can generate anything and everything using this particular package, right at using simply by using your mouse, just by a click of your mouse, nothing else. So there's no need for you to install specific R packages which are heavily used for uh, these plotting these figures. There's no need for that. All you have to do is just fill a form, click on submit, and boom, you are good to go. You have all these plots, all these data inputs available in front of you in your system. Again, what you get access to with your genome stations, it is obviously a subscription based platform which we call as Gstack. Now the problem with current storage options that are available with us, for example Google Cloud, if I Google Drive, if I have to talk about, is that they charge you for every GB of data that you upload, right? You constantly have to keep a track of amount of data that you are uploading. Also the when we talk about genomics data, there's no segregation here. For example, you cannot you have to manually segregate your data, create folders and segregate them into folders pertaining to your raw data, to your assembly files, to your differentially expressed genes, to various plots that you are going to plot using your tools, right? So we have designed this Gstack platform in a way that it also doubles up as, apart from being a dedicated genomics cloud wherein the structure of the entire cloud is designed in a way that you can very easily segregate your data on the basis of, you know, the nature, on the basis of its nature. You can share and collaborate with various users of Gstack quite easily. Apart from that, you can keep a track of your project, the various stages that your project is in. You can very easily keep a track of it using Gstack, Gstack's project tracking features as well. So this is what it roughly looks like. You can keep, and you can keep a track of total space that you have utilized. You can keep a track of various projects and the amount of data that you are utilizing. So not just sharing of data, you can also keep a track of your analysis and the overall profile of the cloud storage that is allocated to you on the basis of your subscription. So this also comes as part of your Genome Station experience. Gstack, a dedicated platform for storing and retrieving and keeping a track of your genomics projects and your genomics big data. Now, if we talk about various Genome Stations installations that we have done, so in a span of around 5 years, we have been able to set up bioinformatics facilities and labs powered by Genome Station in various research institutes funded by several national level funding organizations. The count is in excess of 100 individual units, and these includes these include smaller desktop level variants as well as big rack servers as well. These premier institutes include universities and research centers like JNU, ICGEB, DU North Campus, IBSD, Junagadh Agriculture University, RCGEB, etc., etc. So, and to add on to this, the concerned the users, the Genome Station users, they are not just restricted to a particular domain. Like it's not like they are only working. We have only supplied Genome Stations to people who are working on transcriptomics or animal systems or plant systems or a particular system as such. These the users of these Genome Stations are as diverse as they can get. You name a system which scientists are working on, and you will find a Genome Station set up in a similar sort of a facility. So yeah, right from RNA-seq data analytics to eukaryotic whole genome sequencing, scientists working, our clientele includes scientists working on different genomics-based applications altogether, making it an all-purpose machine that simplifies your data analysis task. Now, this is a small chart of various Genome Stations and the installations that we have done in the past. So we are slowly and steadily we are planning an expansion into the unventured territories as well, and by the end of this year you would see a lot more areas being covered in red. Now I like to give you a small demo of how exactly one such automated application, that is SQIT, works. If you look at the video here, this is the login screen. Okay, so You can register yourself. You can register yourself as well. I have this demo starts from the login process, but if you are a new user, you are required to register yourself first. So once you register yourself, you will be allocated a login ID and a password, which you have to enter here in the login screen. Once you log in, this is your home screen and the various applications which are available for your pursual.
அடுத்து இந்த ஒரு ஓடிட்டு இருக்குது Bionivet workstation. சொல்லு
You can
வாங்க உள்ள வாங்க. அட வாங்க.
you can
இருங்க
start with raw data quality control. You can start with RNA-seq data analysis. You can work on RNA resequencing data analysis. You can work with metagenome 16S RNA data analysis, and there are several other applications as well, right? Now, if I take this further,
ஓவராலா ப்ரோசஸ் பார்க்கறீங்க. நீங்க வாங்க. இத பாப்போம். வேற எதுவும் தட்டி விட முடிஞ்சா தட்டி விடுங்க. எடுக்க முடிஞ்சா எடுத்து விடுவோம். நான் பிரிண்ட் எடுக்கட்டுமா? பிரிண்ட் எடுக்கறேன். PDF
just a second, sorry. Yeah. So if I take this further, let us see how raw data quality control, the one which I had showed you previously, is performed using the SQIT workbench. So you click on raw data quality control, you select the type of data that you have. We have selected Illumina here. You select the type of data that you have. You enter your details, as in you enter the project details here. Basically your project name, your basic QC parameters. You select your output folder, give a name to it, right? And if you can spot here, the question marks in front of each parameter here give you an insight into what should be the input, what exactly does this parameter perform, what function does this parameter perform, and what exactly should be the input that you are trying to provide here. So you create an output folder, write your sample names, simple form filling, nothing else. You select your raw data, which is in FASTQ format, the forward and reverse read if it is paired end, just the forward read, just the single read if it is a single end sequencing approach. And you click on submit. So as you can see here, the analysis, if you go to the current instances window, you can keep a track of your project, the start time, the end time, what is the status, whether it is in process or it is complete. As is being shown here in the demo, once it is complete, you can click on the next step and you can directly go for report generation as well. Okay. Yeah, so this is the report generation protocol. Again, a small form you need to fill bearing in all the details associated with the project. Once you fill that up, all you have to do is click on generate report and you will get a customized report specific to your data bearing all the figures and information that you would need to interpret the overall quality profile of your data. So as we select our output folder here, and we click on generate report, so we get this report in PDF format that can be downloaded as well. So rather than you investing your time in learning Linux, getting to know how to type in the parameters, what does each parameter perform. All you have to do is fill up a form, click on submit, and you will have comments regarding the data and various other figures associated with the data right in front of you in a report format for your interpretation, right? Makes analysis quite simple, isn't it? I'll show you a demo report here as well. That would give you more understanding of uh, the kind of interpretations and that you can generate using a Genome Station. So if you could excuse me for a second. Yeah. So if you can see the screen here, so this is a demo report which we generate. So you will have information regarding the samples here. You will have introduction regarding the pipeline that has been used, right? The various tools that were utilized in this entire workflow. You will have information regarding that. Then you'll have information about the steps, individual step by step, which you can refer from here. So data QC, you talk about the tool, you get to know about the various parameters that were used to process the data, the overall QC profile, that total number of reads, how many of them passed QC, what is the GC percentage, how many of reads were of high quality, how many of them were of low quality. Every information can be gathered from here. Then when you talk about alignment, this is a resequencing screen share is not happening. 1 second. Yeah. So yeah, so you get all information about your samples. You get insight into read normalization, as in if your sample has excess depth, you are normalizing it using various protocols. Then you will have information regarding the alignment tools that were used, what are the overall alignment stats, how your reads have aligned, how many of your reads have aligned against the reference, how many of them remained unaligned, how many of them are multi-mappers. All that information you can generate here. Then this is the graph for the same, and this is sample wise distribution in terms of a table in a tabular format. We also make sure that we provide you with overall remarks regarding your overall data. If at each step we provide you with our specialized remarks regarding the data, how good is the overall data quality, how well is the data aligning against the reference, all this information we try to provide you with expert remarks on the basis of our experience. We have added that part also in the tool here. Then when it comes to quantification, tools that were used, so this report deals with RNA resequencing data analysis. I'll show you one more report for bacterial genome sequencing data analysis as well. So to find out correlation between replicates, you have the principal component analysis here, which we have plotted. Then heat maps to check sample to sample correlation. When it comes to differential data analysis, we have plotted the same using volcano plots for you to get an idea of how many genes are upregulated, how many genes are downregulated. The same can be reflected through this table as well, and you can arrange your genes on the basis of an increasing or decreasing log to fold change value, right? So we give you remarks as well, what all genes in your sample are upregulated, how many of them are downregulated. Everything is covered here. Then a heat map for the same, we have plotted here the heat map also. So you get heat maps as well. Generally we struggle plotting all these extra plots, right? Because most of them are R based. Somehow, if we figure out how to analyze using these complex commands and pipelines, the next step is to interpret those results and depict them using various pictorial representations. So that is a big challenge most of the times. So this pipeline that we have pre-installed solves that issue as well. mm, yeah, again, when it comes to significant biology and gene enrichment, so various pathways, cellular components, various pathways in which your genes are involved, your various gene ontology, basically biological processes, molecular functions, cellular components, everything can be charted out in this single report, right? So for the same, you have for top 5 geoterms, you have these dot plots, enrichment maps, gene concept networks to show relations between various functionalities and the genes which are involved in regulating those functionalities. So you don't have to separately do all of this. All of this is done just by clicking on a submit button, and you get a complete end-to-end analysis for your sample, for your RNA-seq sample right here. We also provide you with methodology, so that if you are including that in your publication, you don't have to struggle for that as well. We provide that as well, that also in this report. We provide you a map of your output directory, as in where all the output files are generated, and a small reference of the publications, citing various tools involved here, and a small acknowledgement also. So this was all about the reports which we generate using this particular platform. Now let me give you a small demo of our cloud storage platform, which is Gstack. So just give me a second. Yeah. So this is how the Gstack interface looks like. You have to enter the login details here in the user ID and password section. Once you enter those details, you have to enter the password. Once you click on login, this is your interface that you are led to, and this interface will give you all insights regarding the various projects that you have loaded in your allocated space, the size that they have occupied, the percentage that they have used, the stage till which the analysis is complete, various applications, and the amount of data that each of that application is utilizing. Everything gets covered here. So this is your project management platform, wherein you get to keep an eye on what stage your project is into, how much data is each application occupying, so on and so forth, right? Also, see, so here we are talking about the various stages that are involved in your, the typical stages that are involved in your project, and what all have been completed, what all are going on. So you get an insight into that as well. Now, if this is all about the project management platform, now if I have to give you an insight into where your data is stored, well, this is how your data storage platform looks like. Data storage interface looks like. Segregated folders for each of your data sets, be it the raw data or the QC based files. You can upload one in one underscore QC folder. Once you're done with assembly, you can upload it in two underscore assembly folder. And depending on the kind of analysis that you perform, you can upload the same in each of your subfolders. Also, if you want to share your findings with someone, if you want to add collaborators as well, that can also be very easily done, because that is the true uh, power of any cloud based data storage platform, that you can share your data on the run with anyone whom you would like to do that with. Okay, so this was a quick demo of Gstack. Now, as I said, we have installed genome stations in a lot of reputed labs and facilities. This is a small feedback that we got from Dr. Shailendra Goyal's lab. Uh, just I would request all of you to just have a look at it. Was it audible?
Yes, you can.
Okay.
Genome Station. I'm from University of Delhi, Department of Botany, working as a research scholar. The broad area of research in our lab are molecular genetics, crop improvement, and genome organization. As you all know, this kind of work generates huge amount of NGS data, and for the analysis of same, we procured this small but very powerful instrument from Bionivet Technology Private Limited in 2020. Array of tasks it performed are pretty very impressive, from small tasks like local blast to the big tasks like complete transcriptome assembly and DEZ discovery were very successfully performed by using this. Big genome data for comparative analysis were also achieved by this instrument. Also, as a user, I personally feel that this is a good addition to our lab, which has saved a lot of time because it had its pre-installed softwares and packages which are most commonly used in our research area.
So this was feedback from Dr. Shailendra Goyal's lab, and this is a desktop variant of our Genome Station. We have workstations as well as tower servers, what we call as tower servers also, and rack servers as well. So depending on the sample load, you can procure that. Now, on as parting notes, I have with you a small framework for an ideal facility, that if you are looking to set up a dedicated genomics uh, data analytics facility, this is how it should pan out as. You should have at least 5 individual nodes, so that the research groups associated with that particular lab can access those nodes. Preferably 64 GB RAM with Core i7 processors. Then you should have a centralized rack server, which would be connected to all these nodes, so that if any of the groups is working on a heavy uh, data analysis project, then instead of running it on their smaller nodes, they can designate it to the rack server. Now, the configuration of that server can vary. You can have a minimum of uh, 256 GB RAM. If you're working on de novo projects, then it should be definitely higher, because if you have to process a de novo genome, then the amount of RAM required, the RAM that you have in your system becomes quite important. So if I give you a straightforward calculation, 1 GB of RAM is required to process 1 million reads. 1 GB of RAM is required to process 1 million reads. That is a straightforward calculation here. So whatever is the amount of reads that you are looking to generate, you can you know uh, contemplate based on this stat. So for de novo experiments I'm talking about, so you'll require a heavy RAM setup when you are looking to analyze eukaryotic genomes. Even bacterial genomes also, if you are looking to assemble, you need to have a minimum 128 GB RAM setup. So this centralized rack server will be connected to all these nodes, so that, as I said, in future, if any of your research groups is looking to work on a heavy duty research genomics based project, then it can be designated to the servers. Then you can also have a network assisted storage, which is also known as NAS. It can act as a centralized storage facility, wherein data from the server as well as all your individual nodes is stored, and that NAS can be backed up onto the cloud server, which is Gstack, so that you can keep a track of your project, how well it is proceeding, and what are the stages that your analysis is into. and you can easily collaborate and access your data from anywhere. Also, what is recommended, what is recommended here is a centralized power backup for the entire facility, because voltage fluctuations can damage the SMPS of your systems. Now, once this is done, I'll give you a short what I'll call it as the ideal processor RAM combo for your systems. So if you are into resequencing 2 to 4 samples, a minimum 32 GB RAM, 4 cores, 8 threads, 4 to 8 samples. If you are into resequencing, 64 GB RAM, 6 cores, 12 threads, 8 samples and beyond, minimum 128 GB RAM, 12 cores and 24 threads. Similarly, if you are looking to analyze de novo projects, then lower eukaryotes, 2 samples, 32 GB RAM, up to 6 samples. I think 6 is also too much. Up to 4 samples you can analyze in 64 GB RAM. And if your sample load is 4 samples and beyond, then you ought to work or set up a facility which has a minimum of 128 GB RAM and beyond. Now, this is all about it. So I would like to end my talk here. Thank you so much for your attention.